Okay. So, are you seeing my screen now?
Yes.
Okay, perfect. Okay. Then, thank you, Amber, for giving me the opportunity to talk at the Virtual Monthly Lab meeting. And as the title say, so today I would like to talk to you about how we characterize the human GAT1 or SLC6A1 using solid supported membrane electrophysiology and automated patch clamp. So we are more on the basic science here, if we want to say that. So I divided the talk into 4 sections. I would like to start giving a general introduction on the protein itself, focusing more on the mechanism of action of the protein. Then I would like to speak about the techniques that we use to characterize it. Then we will see the validation of our sample using solid supported membrane electrophysiology and automated patch clamp. And in the end, I would like to share some data that hopefully gives some new insight into how the protein works. Okay, so let's start with the introduction. Here, I guess that for most of the people here, there is nothing new. So the protein is part of the SLC family, so it's more than 400 members in this protein family. It is the main function of this protein is the re-uptake of the neurotransmitter GABA from the synaptic cleft inside the presynaptic neuron once the signaling is over. It is mainly found in astrocytes and neurons in the central nervous system, and it is a secondary active transporter, meaning that in order to transport GABA back inside in the presynaptic neuron, it needs to use the sodium gradient to energize this reaction. And of course, it is a very important also pharmacological target because, as again you well know, dysfunction in this protein are related with severe neurological disorders. Okay, so here just let's just a brief have a brief introduction of how this protein works. So this would be GAT1. This would be the membrane of our neuron. This is the inward compartment, or basically the neuron cytosol. This would be the outward compartment, so basically the synaptic cleft. And importantly, we can see that we have a lot of sodium, so these yellow spheres here. We have more sodium ions in the synaptic cleft with respect to the inside. So this is our sodium that will energize the whole transport. So we start by having the protein in the outward open conformation. So the protein is outward towards the synaptic cleft. Then the protein will bind its substrates. Those substrates will be 2 or 3 sodium ions. There is still a little bit of debate in literature if the sodium ions are 2 or 3. Then there is the support of a chloride ion, and then we have our GABA molecule that binds inside the protein. Then the protein undergo a conformational rearrangement, a fast conformational rearrangement, so it goes into an occluded state. So in this particular state, no more GABA can bind the protein. Then the protein opens towards the inward compartment, the cytosol of the neuron, and releases its the substrate that were bound. For chloride, there is still there is still a little bit of confusion here. So some says that the chloride is actually released. Some says that chloride basically kind of bumps in and out and stays in an equilibrium with the protein itself. Either way, the protein then reverts back to the outward facing conformation, and it's ready to start a new cycle. So now let's talk a little bit about the techniques that we used. So there are plenty of techniques to study transporters. We focused on electrophysiology. So basically what we need is, of course, a current to be generated by those proteins. In particular, basically, we need that what is an electrogenic transporter is a protein that moves charges in a way, so moves molecules in a way that there is a net charge that crosses the membrane, so we can read the current. Yeah. Luckily, GAT1 is an electrogenic transporter, so we can study it with electrophysiology. So we can start with the patch clamp, automated patch clamp. So we start with a cell that has GAT1. Here in this short video, you can see that the cell is attracted to a hole via suction. Once the cell reaches the hole, then again with suction in the cell membrane, and at this point we have the cytosol of the cell comes in contact with the electrode that is here. Electrogenic reaction happens here, can be read by the electrode, and then we can have an electrical readout. So here we use the patch liner from Nanion to perform this measurement. Here is a short video on how it works. So we have a well, we fill it with a solution, then we let cells go in. Eventually, one cell gets catched into the hole, then the cell go back, the other cell will are flushed away. We can start the recording. So in this case, we have voltage control, so we can apply, for example, voltage jumps and see what happens to the transporter or the channel. We can add compounds and again see what happens. In this case, important in this case is that we have voltage control in this case, and we are working with whole cells. For the solid supported membrane electrophysiology, basically we need to elaborate our sample a little bit, so we don't use whole cells in this case, but we have to break our cells to obtain a more pure, let's call it, sample. So we can proceed in two ways. So we can break the cell and then purify membrane fragments that have the protein of interest on them. And then those fragments will reform vesicles that have our protein of interest. Or we can, again, break the cell, then purify the whole protein, add lipids, and in this case, we will have the lipid reconstitute with the protein in a structure, in a vesicular structure that is called proteoliposome. So the difference between these two is that in this case, we still have some of the native protein that are present on the cellular membrane, while in this case, the protein is pure. So we will have our just our protein on lipids. Either way, we can use solid supported membrane electrophysiology to measure this kind of sample. So here is the high throughput system with a 96 plate. Here is the lower throughput system that can just measure one sensor at a time. So how does this work? We have a sensor, which has a layer on top. And on top of this gold layer, we build an artificial membrane composed by a tile layer and a lipid layer. Then we pipette our sample, so our vesicle or proteoliposome that contains our protein of interest. Then we put the sensor in our machine, so now we can have an electrical readout system. And then we perfuse with the substrate. So in this case, it's important we don't have voltage at the beginning of the measurement, and we don't have voltage control in this kind of technique. So everything is driven just by concentration gradient of the substrate. Either way, now the transporter can get into motion, and charges accumulate inside the vesicle. Since the membrane, this double membrane system forms a capacitor, then we have that the sensor can react to the accumulation of charges inside, and we can have an electrical readout in the end. This is how our SSME normal workflow looks like. So basically, we have a continuous perfusion of three solutions. So we have a non-activating solution that does not contain the substrate of interest. And this is useful because we can get rid of the mechanical artifact that rises up when the fluidics is into motion. And also we get the baseline for our measurement. again, this non-activating solution can anyway contain accessory ions that can establish the needed ion gradient, for example, for GAT1. Then we perfuse with the activating solution, and in this case, our activating solution will contain, for example, GABA, and this will generate, hopefully, a response from the instrument. And then again, we want to wash out everything and go back to the initial conditions, so perfuse again with a non-activating solution. And the system, after basically going in reverse, will go back to the initial conditions. So here it's just an overlook on how the current from transporter can look like in an SSME environment or in a patch clamp environment. So if we are looking at a transport event, this is a dynamical equilibrium. And in SSME, we will see monophasic decaying current, so a slow decay that just decays with a mono-exponential function. In a patch clamp environment, we will see a rising phase and then basically a steady state, so a straight line for our current. Then we can also have pre-steady state events. So those are out of equilibrium events, and those are usually associated either with the substrate binding or with a conformational rearrangement of the protein. For example, the occlusion of the protein could be one of those. In this case, we see that between patch clamp and SSME, we have analogies. So we will always see fast transient currents. Or we can have a more complicated output and having, like in this example, we can have basically a combination of the two. So like a fast pre-steady state current and a slow decay transport. And in the case of patch clamp, it would be again fast transient pre-steady state and straight line for the transport current. Like here. Okay, now let's see how we validate our samples using solid supported membrane and automated patch clamp. So we start with the automated patch clamp. We have a cell line overexpressing human GAT1. It's a Chinese hamster ovary cell line. So we throw those cells into a patch liner and we hold the potential to 70 millivolt, which is the resting potential for neuron, and then we perfuse GABA for 12 seconds. As you can see, we have a response, a current response that is defined by different GABA concentration. So what we did, was we plot the current as a function of the GABA concentration that we used, and from this we can obtain an EC50 or Michaelis-Menten constant, which is an important kinetic parameter. So it's the concentration needed to reach half the maximal current, which is basically half the maximal speed of the transporter. And this, and the value that we obtained is in very good agreement with the literature results. So we're confident that our cells were working nicely. Then we switched to solid supported membrane. So we broke our cells and we reconstituted membrane vesicles. We filled our vesicles with potassium chloride. Then we started perfusing a solution that was containing only sodium chloride. So what we obtain now is just the chemical gradient of sodium that wants to go in, but still nothing is moving here. So we just have this potential force given from sodium outside. And then for another second, we perfuse a solution containing, again, sodium and GABA. So this time we have our main substrate, the transporter is set into motion, and we can read the current. So what we got out is using CHO, using our vesicles containing GAT1 in presence of 150 millimolar sodium chloride and an enormous quantity of GABA, we could see a very nice current from here. while as a control, we used vesicles without GAT1, so empty vesicles. We treated them with the same solution, so GABA and sodium, and we didn't see any current going on. And then here I just did an accessory test. So basically, I wanted to see that what the current was actually, so that we needed sodium and chloride to have these currents. So I took again our vesicles with GAT1, perfused with a solution without sodium and without chloride and GABA, and again I didn't see any current. So we also did some very rough pharmacological validation using, again, solid supported membrane electrophysiology. We used this very well-known GABA transport inhibitor, SKF. So here we analyzed the transport component analysis. We will see later why I picked up the current here instead of here. But anyway, this is the transport component, and again, if we plot the current as a function of SKF concentration, we can see the potency of this inhibitor, and this value is in very good agreement again with what it should be. So now, just to see a bit of data that we were able to generate with solid supported membrane electrophysiology. First of all, as the title says here, we see 3 electrogenic events with the solid supported membrane electrophysiology, while in patch clamp we only saw 1, so we only saw the straight line basically for the transport. So if we have our vesicles that are filled with potassium chloride, and then outside we perfuse with high sodium and high GABA, so now we have a very strong sodium gradient force and also very strong GABA concentration gradient, then we observe a current that is exactly the same as I showed before. And this current has 2 components. So we have a peak, E2, and then we have a slower decaying phase here that we call E3. Then we wanted to see the effect of sodium gradient. So basically what we did here was to fill the vesicles with a lot of sodium. So we have 145 millimolar sodium chloride inside and 150 outside. So now the sodium gradient isn't very strong. We perfuse with a lot of GABA, and now we obtain a current that is like this. So we have again this very slow decaying component, E3. And now we have a very fast spiky current that we call E1 that is different from the other because the decay time is an order of magnitude faster. Then in the end, we went back to having a very high sodium gradient. So again, no sodium inside, high sodium outside. This time we perfused with a small quantity of GABA, and in this case we were able to resolve 3 different events, which actually were the same events that we observed before. So it's just the sum of the 3 events in a single current. So we can see the fast event E1, the slower event E2, and then the even slower event E3. So we try to attribute some identity to these components. So we think that the first component, E1, is the occlusion step of GAT1. So when the protein is outward open with everything bound inside and then it closes, we think this step is electrogenic and generates this fast spiky current here. The second event is a little bit more tricky. We think this can be a sodium leak state of the transporter. They have been reported in literature, and this would also account for a discrepancy in the stoichiometry that have been observed. So basically, it's possible that the transporter, even if occluded, can generate kind of a channel through which the sodium ion can pass. For the third component, we do think this is transport because I read the current here with different GABA concentration, and if I plot this thing. Again, I can obtain the EC50 in the low micromolar range, very similar to what we also observe with patch clamp and very similar to literature. So we think that this is actually transport. Also, the fact that it's blocked by SKF and gives us confidence that we are looking at this. Then we wanted to try and shed some light on this sodium GABA stoichiometry. So we performed stoichiometry assay. I will share you the physics here. Just knowing that we are at 0 millivolt at the initial of the recording, lets us say that the free energy associated is just a function of the stoichiometric coefficients and the substrate concentration, so GABA and sodium concentration. So we played around with those concentrations. Those are the example traces from the experiment. And if we plot the total charge as a function of this ratio between the concentration, we have that when the charge is 0, this ratio is equal to the stoichiometric ratio between sodium and GABA. And we see that this nicely goes to 2. So we think that the sodium GABA stoichiometry is 2 sodium for each GABA molecule that is transported. Okay, so as a summary, solid supported membrane electrophysiology seems to be a suitable electrophysiological technique for measuring GAT1 currents and also to perform some pharmacology. It also sheds some light on the fact that still the GAT1 mechanism is not yet completely understood. And so it needs further investigation in basic science in order to then understand better how this protein works and hopefully have better and more useful drugs to treat eventual disorders. And then in the end, again, functionally speaking, we confirm that sodium GABA stoichiometry appears to be 2 to 1. So it's all, and I thank you for the attention. If there are questions, I'm happy to take them.
So, oh, go ahead, Mike.
So Rocco, thanks. It was a nice talk. Can you fill in anything about what you think is going on with the sodium leak? And does that not appear in the stoichiometry, in the thermodynamic calculation?
Yes, sure. This is a good question. So it doesn't happen. So we don't see it in the stoichiometry assay simply because the sodium gradient here between in and out is not very high. So as you can see here, the maximum difference between in and out is 14 millimolar sodium. And this I have data on the fact that we don't see this event issue in presence of such low sodium gradient, basically. So that's why I think the stoichiometry data doesn't reflect this sodium leak. About the sodium leak itself, again, I say this is a theory mostly. The only thing is that an event that occurs after after all the substrate are bound to the protein, and basically before the protein itself is switched to the inward facing. So it has to be somewhere in between, and that's why I think it can be actually a sodium leak. It is also it is dependent on GABA concentration. It is dependent on sodium gradient and on sodium concentration. So that's why I think it can be basically a substrate-driven sodium channel behavior of the protein. I hope this answers your question. Yes?
Yes, thanks. That answers the question. Do you think that it's sodium selective, or do you have any data addressing that, the leak?
So, sorry again, I kind of broke up a little.
Do you think that the leak conductance is selective for sodium?
That's another question that we need to address. But from literature, I know that lithium can also have this kind of effect. So I wouldn't say that it's very selective, to be honest. But of course, this needs further investigation.
Okay, Dr. Radish, did you have a question next?
Yeah. So I guess the idea then is you want to use the solid supported membrane instead of the automated patch clamp. Is that correct? That's what I sort of get the idea. You're
Yes.
going towards that way. You don't want to combine the two. Is that right?
So no. The point is that, of course, both of them they can give how do you say, complementary information, because with solid supported membrane you don't have voltage control.
Great.
Which of course we are talking about a neurotransmitter transporter, so we have a voltage dependence, and it's of course very useful to study that too. The problem is that yes, please.
Did you validate your measures with the solid supported membrane EFIS at 0 millivolts with the automated patch clamp?
No, because that was not possible. With patch clamp, basically the experiment that I performed here is just because we have chips with 4 holes, so I was able to record 4 cells at a time, given this signal. So this is the sum of 4 cells. If I tried with a single cell chip and I didn't see anything, even at minus 70 millivolt. I also tried with the 4X chip at with the 4X chip at 0 millivolt, and I didn't see any current. So it becomes too small to be detected.
Sorry, more technical questions. And the internal that you use with the automated patch is similar to what you did with the solid membrane.
Yes. So
you don't need fluoride in the automated patch clamp to get decent.
No, in this case, yes, I wanted to add that in this case we need fluoride anyway to establish a better seal. But as of now, I think we have also fluoride-free measurements. Anyway, in this case, so I would say patch clamp, I used it. It's really not my strong expertise. So I'm more focused on the solid supported membrane electrophysiology. But in this case, yes, I used fluoride to enhance the seal.
And the fastest time base that you can get, are these always going to be in seconds, or you can get faster than that?
So for patch clamp, I perfused GABA for 12 seconds, while for SSME, just 1 second. So of course, the temporal resolution is way higher with solid supported membrane electrophysiology. This is also why we can detect these different phases in the current. So the point is that, of course, if your temporal resolution is not high enough, you tend to see a current that is a sum of everything. While if the temporal resolution is high enough, then you can actually see, you can resolve the different contribution.
Thank you.
You're welcome.
Okay, we have some questions in the chat. The first is, go ahead.
Go.
Can this method be done with dissociated neurons?
So the solid supported membrane electrophysiology, I mean, we tried some whole cells also on these devices. We have some results, but it still needs to go through thorough testing before saying that it's safe to use whole cells with this technique. So there are ongoing measurements for that. So hopefully in the future, if we can if we tweak that out, we can also be able to use neurons. Of course, I know that culture neurons is very difficult, so it would be quite a challenge. I can guess.
Okay, and has anybody looked at is that chloride or sodium?
Chloride.
Okay, I think it's sodium.
No, chloride.
Dependence.
Yes.
Oh, okay. Dependence of
I mean,
GABA-1.
it can be chloride also. I mean, it is dependent on both.
Okay.
So I didn't show it here, but I also performed the stoichiometry assay with chloride. But in my hand, it doesn't look like chloride is thermodynamically coupled. I would say that in past experiments, it has been shown that if you are in complete absence of chloride inside the vesicle, then you can clearly see a coupled transport of chloride. But as soon as you have a consistent amount of chloride inside the vesicle, then this is not clear anymore. So basically, I would say that the main function of chloride seems to be basically to counteract one of the sodium charges in order to stabilize the whole system. Because one thing that has been discussed in literature is that the electrochemical gradient of chloride is basically almost in equilibrium. So there is no, let's say, driving force given by chloride being transported that could help, let's say, the GABA transport, while this is true for sodium.
Okay.
I hope this was the question.
I think so. Since the patch liner was able to detect the GAT1 signal, do you expect the synchro patch can also detect the small signals?
As far as I know, so I would say that if there is the way to have 4 holes in each of the synchro patch, well, maybe yes. But if there is only 1 hole, I think that the possibility are very, very, very small. Because again, you need to have a lot of protein to detect it with patch clamp, because the transporters are slow, and patch clamp, like temporally wise, basically, patch clamp works well with channels that are faster, and for such things as slow as transporters, for now, solid supported memory is better. But anyway, further development could happen, and yeah, would be nice anyway to see that happening also.
Wonderful. And Henry, I think you're next.
Thank you, Amber. Thanks, Rocco. It's a very interesting presentation. I'm wondering, in your essay, can you look at a population of transporters? If you have some normal functioning transporters, but some are mutant, can you tease out that difference? And if you have, for example, a gene therapy situation where I replace a functioning transporter, will you be able to use this method to tease out the difference and the recovery?
So you mean if I am able to assess how many transporters do we have?
Right.
No, as of now, no, it's not possible. So basically, the problem is that having this capacitive-coupled system, we have that all these currents, they decay, but when they decay, they also decay as a function of the sensor itself. So it's like kind of a mixed kinetics, and therefore, with the technique itself, it's very hard I would say it's not really possible to say how many transporters do we have. So for that, yeah, you would need something else. Exactly, like, I don't know, like fluorescence readout of whatever is on the sensor, or something similar.
Right. I'm just wondering, because in your some of the results, you show that there are different phases of that decay, and would that be reflecting the functioning of each transporter per transporter?
So this holds true for, just let me go back to the traces.
Yeah.
So this holds true for this one, so for the fast one.
Right.
Because in this case, probably this decay is not a function of the sensor itself, but it is an intrinsic kinetic parameter of the protein itself. So in this case, of the occlusion, or what we think is the occlusion. Actually, I studied this decay, and we think that studying this decay, it's possible to get the KD. So basically, the thermodynamic affinity constant for GABA binding. Having said so, for the other two events, so for the E3 event, no, because this is already in the range at which basically we have filled the membrane capacitor. So now basically we are in the discharge phase of the capacitor, and so now we have the physics of the capacitor itself coming into play. So now the kinetic is completely mixed, and this cannot be used to say nothing about the transport phase.
Thank you.
Okay, Dr. Rogowski?
Yeah, thanks, Amber. We're bumping up against time here, so I'll try to make this quick. From a clinical and translational perspective, Rocco, things that you might want to use an analysis of the GAT1 transporter for, firstly, would be understanding how a variant might affect the transport activity, or affecting how a medication or treatment might affect GAT1 transporter activity. And in the past, people have used biochemical methods to assess the transporter activity, and you mentioned conventional patch clamp. And what I'm interested in knowing is, where would the SSME methodology fit in? How would it provide improvements over the conventional approaches that people have used?
Well, mainly because the fact that you can detect different events. So, like I showed here, so these triplets of event you cannot see with patch clamp, since the time resolution is different. Also, the fact that SSME intrinsically grants an amplification of the current itself, because we have so since in patch clamp basically you rely on what happens on just one cell. In this case, you have a gigantic amount of transporters that are adsorbed on top of the sensor. So there is also an amplification factor. And
so in a
I
way,
would say that
the analogy I would make, Rocco, is with gating currents. So you could use patch clamp to measure, for example, for a voltage-dependent ion channel, the current, but you can also look at gating currents that would look at the conformational change that occurs when the voltage change is imposed. So I gather that's would that be a valid analogy?
That's basically okay. That is a current. That's a good point. That is a current that doesn't appear here, because that is so this pre-steady state current that is also observed, that is actually observed in patch clamp or 2-electrode voltage clamp, to be more precise, and not in solid supported membrane electrophysiology, is another pre-steady state current that is completely driven by voltage. So in this case, we don't see it with SSME, we see it with 2-electrode voltage clamp. And this is basically this voltage-dependent flip of the transporter from the inward facing to the outward facing before being ready to receive sodium.
Yeah. So I guess the final thing I should ask then is, have you looked at any variants? And can you characterize the changes in these various signal components?
Unfortunately, no. So this is so there is still plenty to do here. So we should look at other compounds, so also even maybe enhancer or other drugs, and also check, of course, some mutants and see if they show actually differences here. But unfortunately, we could get hold our hands on such samples.
Great, thank you.
You're welcome.
Kirill asked, how about liposomes extracted from tissue used in SSME?
Sorry, again?
Okay, I'm going to re-put it into the chat because maybe I'm saying it incorrectly.
Yeah, I can stop share because otherwise I cannot see the chat.
Yep.
I can just ask it. Could you take liposomes from tissue and use those? Or would you think they're not going to be, well, first of all, would that work with the setup, and secondly, do you think there just would not be enough GAT1 because you're not overexpressing it? What do you think it could work?
So liposome extracted, I have no real knowledge about this liposome extracted from tissue. So you mean we extract so it's not proteoliposome, so it's not this purification of the protein and their reconstitution in lipids, but this is directly from tissue.
Yeah, because I know in our lab, for instance, we're able to measure GABA uptake in liposomes, so we can measure radioactive GABA uptake. So there's, there are these small lipid vesicles that still contain functional GAT1. So, but it's extracted from mice. And then you can, of course, compare like a wild type mouse or a disease model knock-in mouse. So I wonder if you could
I never tried them, but I wouldn't say why not. I mean, in the end, the important thing is that you have your protein encapsulated in a vesicular membrane, so in a lipidic environment. So, I mean, potentially whatever has this kind of essence, let's say like this, can be, of course, used on SSME. One point is that, of course, it's usually preferable to have overexpression to some degree, but still, one can also try with native. There is nothing against it. Maybe just the fact that if the protein is not expressed enough, you don't see any current.
Right, that'd be what I'd be worried about. And I wonder, I mean, I suppose one could also target brain region like striatum that has a lot of GABAergic cells, hopefully amplify the amount of, well, I guess you'd get GAT3 in there as well if you do that. But,
that of course would be nice, just to have access to a lot of different samples. So unfortunately, this comes basically with my PhD study, and we only could have access to this cell line overexpressing at once. So I did everything with that. But of course, again, if it has a lipidic environment and you have your protein expressed, then you can just try and put on the SSM sensor and see what happens, basically.
But with the whole cell patch clamp, you were mentioning that basically with the 4 cells you were patching, you're just getting enough current to make the whole technique work. Or even doubting whether or not I think screening different variants would be viable because the sensitivity is not quite there.
Also in that case, I mean, exactly. So in patch clamp, I had to use this 4x. So basically take the current from 4 different cells, because if I tried with one, I couldn't see anything. In this case, there is 2 electrode voltage clamp, for example. They use oocytes, so those are big cells, and they can have a lot of transporters on top. And in this case, also, in this case, the current will be amplified quite a lot. But yes, in patch clamp, you need a way to amplify that signal. That's the point.
So in patch clamp, basically, you are just getting enough, overexpressing and using 4 cells. So for the SSME, did you feel like you had kind of a larger amplitude to where if that signal significantly diminished, like let's say if you did use liposomes from tissue, where you'd still be able to detect something? Or you also think that in your setup, when you were overexpressing, you were basically still just at the threshold where you're just detecting, and if the expression went down, it wouldn't be functional anymore?
Well, I have to say again, with patch clamp, since with another expressing system, I struggle to see some currents, and this is, of course, this holds true for transporters in general when talking about patch clamp. Of course, using native tissue, I would expect that if you are lucky, you can get some current, but it will be very, very small.
Yeah, thank you. It was a really interesting presentation. Thank you very much.
Thanks.
Okay, I think Angela had a follow-up question. It's in the chat for receptor-mediated currents.
Where are we? Okay, here. It is possible to measure receptor-mediated currents with SSME. Yes, of course. I mean, you can also see channels with SSME. So anyway, it's just whatever. So if you have a flow of ions, or anyway something that generates a current that crosses the membrane, and a detectable current, then you can see a readout from the instrument. Like we have, for example, TMM 175 is a channel, and well established with SSME. So there is, as long as it's electrogenic, let's say, you can see it with the technique.
Okay, I think Elena. Oh, okay, Elena just asked a question, and she just commented her questions have been answered.
Oh, okay. Yes. Okay.
Does anybody else have questions? Okay, wonderful. Well, thank you so much for the presentation today. It's a great discussion, ran over, and I will make this video available to everybody.
Thanks.
Have a great Thursday, everybody.